Our next speaker is Dr. Peng Li, a Cancer Immunology Scientist at Eisai of Japan. And Dr. Peng moved from his home in China to Japan in 2010 to pursue his education and obtained his Ph.D. at the University of Tokyo in 2017, where he studied immunology of inflammatory bowel disease at the Department of Applied Biological Chemistry. And during his PhD, he went on as a visiting research fellow at the University of Nottingham in 2016 to research chronic obstructive pulmonary disease at the medical school. And following his PhD, he completed his postdoc at Kyoto University Medical School in 2019, working on cancer immunology research in Honjo's lab that won the 2018 Nobel Prize in physiology or medicine. He then joined Eisai in 2019 as a scientist in cancer immunology research. And Dr. Li will be sharing with us their immunogenicity assessment during the development of antibody therapeutics.
Okay, thank you so much for giving me this very honored chance to speak my work. Actually, today I'd like just to talk about how we, in collaboration with between Nobel and Eisai, I can't talk about details on the our project because as I mentioned here, because it is confidential. So this is today's topics. I'd like to give a brief introduction, project introduction, really brief, and then I'd like to talk about immunogenicity issue in our in my project. And then I'd like to talk about Eisai ProImmune in collaboration, how we work together, and finally achieved our goal. And then I'd like to receive your question. Is okay? Yeah. And I apologize for that. For current project, I cannot open any data here, because our drug is is under development. I just give you a very brief figure to show where are now. So currently we already completed the modality selection and compound production. After that, we already fully completed the in vivo efficacy study and the safety study. During the efficacy study and the safety study, we already conduct the another assay, including in silico and in vivo in vitro DCT assay to evaluate the immunogenicity. So already finished the immunogenicity assessment. So currently, after the immunogenicity and other PD or biophysical assessment, we finally select our lead candidate compound, and then we conduct the optimization for the lead compound to select the lower immunogenicity and higher efficacy for our project. So this is a very brief introduction. Yeah. So next is immunogenicity, just mentioned, introduced by Professor Choi. So I really want to skip this this talk, but yeah, just for our I just want to introduce the issue or problem we faced in our project. So we started the current project 3 years ago, and we selected our lead compound, several lead compound. So, however, when we treat the mice, conduct the in vivo preclinical trial by using animal, we found some compound show very high immunogenicity in animal. So we have to seriously consider this problem and to solve this problem. So finally, we start to talk with expert in this field. Like we get some serious advice from the experts. We finally select the lead candidate drug by in collaboration with ProImmune and other partner. What we conduct the experiment, like we evaluated the immunogenicity by using in silico T cell score and in vitro DCT assay. And but also we conducted another PD characterization and biophysical characterization. So anyway, so as we all know that over 90% of therapeutic protein or antibody, they will cause immunogenicity, which may be induce high ADA in patient. However, currently, before the clinical trial, we do not have another approach except including the DCT cells and the T cell in silico T cell score. So because we use an animal, we use a rodent and primate to conduct the antibody for human. It's totally different and partially different between human and mouse and other animals. So to solve this problem, currently I think we think the tool or the approach for us is in silico T cell score and in vitro T cell assay by using human PBMC. So finally, we started to contact with the ProImmune company in UK from last October, I think. Yeah. Yeah. Since we start the inquiry, so the ProImmune should very hot and very quick and polite response for us for our project. So we talked to each other by email and sometimes by telephone or web meeting. So finally, we made the final decision in November, just one and a half, I think, 1.5 months. So after that, we start the shipment and signed the deal. After that, they just I think maybe 6 to 8 weeks for the measurement and completed the the DCT assay on time and shared us the data, fed data on time. So after that, not only she just feedback our data, but also we have, I think, two times, web discussion and follow up by email. Yeah, it's very helpful for us to make the final decision. So finally, we achieved our milestone. We greatly appreciate your cooperation. So in the next slide, this slide, I just show the partial immunogenicity data here. So in the DCT assay, we actually test 11 compound by using the DCT assay. Besides the tested compound, they also add the positive control like PPD and KLH. So finally, we get the data show here, like this positive control worked very well, 100% show the immunogenicity and also show very high strength of response, and the response index should be calculated by this. You can check. Check the. I think the homepage is very. It is have the details for the DCT cell assay. Yeah. So I just want to explain that. So our compound from compound 1 to compound 11. Finally, we get the data. Show a different percentage is immunogenicity and strength response, and finally we calculated the response index. In our experiment, we ordered. I think we used 40 different 40 donors have different HLA type individuals. So for our data, we test 11 different compounds. So for each compound, we had 40 different response for per person. yeah, very good data for us to judge which one have a high immunogenicity or which one have a low immunogenicity. So finally, we ranked all compounds by using the response index, and but also use our in silico T cell score, which was conducted by another partner. So we select our lead compound with this DCT results, in silico T cell results, and our in vitro PD and biophysical data. So finally, we select our ideal compound. So the compound now is under investigation for the efficacy in vivo. So for the next step, we will confirm the safety after the optimization here. Yeah. So yes, I'm so sorry I do not have a very good speak today. Just want to show our great appreciate for your cooperation. Really helpful for our project. Yeah, we successfully selected our lead compound for our project. Thank you so very much.
Great, thank you for that talk. Excellent. We do also have a question in the chat box. So this is from Disha. And this is, does epitome prediction and deimmunization require the knowledge of some languages like R? Does that work like protein docking? That might actually be for our first speaker. Looks like there's
okay, yeah.
quite a few chats going on, but I thought that one was referring to this one. Okay. I don't see any other questions. Are there any from the audience who'd like to ask verbally?
Thank you very much.
Okay. All right. Well, thank you again. Yes,
Emily, sorry. Yeah, I was just going to say, thank you very much, Dr. Li, for your comments. I think I was just going to sort of also just highlight that I guess one of the key things about the dendritic cell T cell proliferation assay is that it's obviously about trying to characterize the potential for T cell responses and how that can then obviously drive unwanted immunogenicity. And we often get sort of questions from clients, such as yourselves, but obviously many others as well, about what's the best way to characterize proteins in that context. And again, it'd be kind of good to have your commentary on here if you can share that experience. But one thing when you have a number of different proteins that you're trying to compare in these assays, understanding that they've all been expressed in a sort of a similar expression platform, so that ultimately they're comparable in these types of assays, is very helpful. Because the key thing, of course, is that you as the client can fully characterize those proteins and the specific characteristics of those proteins. So any levels of impurities, any levels of host cell protein contamination, any levels of aggregation, you will have all that biophysical characterization to hand. And so being able to kind of run these in the assay alongside one another is where it really does add a lot of value. I don't know whether you have any kind of specific input on that in the context of those points. Is that something that you can comment on as well?
Thank you. Yeah, you mean special epitope for that, right?
Yeah, just it's more, I guess, more your experience on being able to run multiple proteins in parallel, because obviously you're having to then produce those proteins and you can characterize them. Were there any sort of specific characteristics that you're looking out for in the analysis of these projects?
Yes. Actually, yeah, we not only conducted not only assess the immunogenicity, but also PD and other in vitro and in vivo. So how to answer your question? I'm sorry, I maybe we can talk about by email or later. Sorry for that.
No problem at all. Thank you. That's great.
Thank you.
Yeah, please unmute yourself. You can go ahead and ask.
Yes, hello everyone. My question is, when we do innate immunogenicity study, what is the concentration of the product which we have to use? We have to go up to as low as high as up to 10x or 5x. What is the expectation from agency based on your experience?
From the ProImmune perspective, Gita, if that helps. So the molecules are typically run in the study at a fixed molar concentration of 0.34 micromolar. And that's equivalent to about 50 micrograms per mil of an antibody therapeutic. That's been empirically determined as being an optimal concentration because it allows us to then compare proteins at a fixed molar concentration. That's not an absolute value, though, of course. So we've certainly done studies where you may be working with a specifically potent molecule, or you might want to look at the impact of overall T-cell responsiveness at different concentration levels. So we can certainly run them at those different levels, but normally we'd run them at a single concentration, and 0.34 micromolar is that optimum concentration.
Thank you. Thank you.
Okay, any other questions? There's some nice chatting going on. Let's see. Okay, we'll go ahead and move on then. Thank you again.